Okay, can you hear me?
Yes, we can hear.
Okay, the screen is visible, the slide.
Yes, yes, everything is.
Okay, okay. Let's talk about the vaccine, which is the last part of my talk. So you must have heard the vaccine from the Oxford Institute, that the Jenner Institute. So there are two vaccine that is very close to phase three, phase two or phase three clinical trial. One is from Oxford and another is from Moderna in USA. So the Oxford Vaccine Institute, Jenner Institute, designed a vaccine is the based of the gene sequence of SARS-CoV-2 spike protein. So the sequence has been inserted into the adenovirus. So what this adenovirus cause? That it causes the cold symptom in chimpanzee. But this virus is weak and cannot cause disease in human. So this adenovirus is a vehicle to transmit the SARS-CoV-2 sequence, coding sequence inside the cell, into our cell. So this recombinant adenovirus carrying the SARS-CoV-2 sequence has been injected into the human body, and this chimpanzee's virus expressing SARS-CoV-2 spike protein will be ingested by the macrophage. So, and will induce the same immune machinery. So, when macrophage will ingest the protein that is being expressed by the chimpanzee's virus, this protein is recombinant SARS-CoV-2 virus, but expressed by chimpanzee's adenovirus. So this will be challenged by the macrophage or antigen presenting cell, display the peptide, activates the T helper cell. T helper cell will transform into cytotoxic T cell, and also it will activate B cell to make antibody. So the T helper cell upon viral peptide recognition will produce cytotoxic T cell and induce B cell to produce antibody. So now this production of antibody against SARS-CoV-2 spike protein by B cell and memory B cell. So this is the beauty of the vaccine. So the vaccine not only generate B cell, it also generates memory B cell. Why? Because this long-lived memory B and T cell will recognize the virus, so they will stay in the system as silent. But when you will be infected by the virus, then they will immediately activate and start making antibody. And this antibody will kill the virus, and cytotoxic T cell will also kill the virus. So there are both B cell and cytotoxic T cell mediated cell killing of the virus will take place if you are vaccinated. So this is the hope. So this is one way of vaccinating. So the another way that is also producing a very good result here is the Moderna's mRNA vaccine. So how this mRNA vaccine technology works? So this is the spike protein S sequence. That means the RNA sequence of the S protein, and this RNA being coated by a lipid or the nanoparticle. So this lipid based nanoparticle will be injected into the human body, and then this nanoparticle will protect, otherwise this RNA will be degraded, because in the cell there is a lot of nucleus is also there, so the RNA has to be protected. So this nanoparticle will fuse with the cell and then will be challenged by the cellular protein synthesis machinery called ribosome, and then ribosome will synthesize viral protein. So by injecting viral sRNA, you can make viral S protein in the cell. And then when viral S protein will be made, that's for SARS-CoV-2, then again same pathway will be activated, that then it will be displayed by the antigen presenting cell, activate the naive T cell, CD4 plus T cell, CD8 T cell, and then that will eventually activate B cell. B cell will produce antibody, and antibody will protect the virus. And not only B cell, so B cell as well as memory B cell, both will be produced. Actually, this technology has been published well before, that they have tested this technology first with rhesus monkeys that shows that activation of antigen presenting cell in vivo by modified mRNA. So this is the first time that we are seeing that by injecting mRNA also, you can synthesize the protein inside the cell and then make the antibody response. So why the result of phase 1 clinical trial of mRNA vaccine conducted by Moderna is highly encouraging. So this has been published like several weeks before that. So each participant, they did a phase 1 trial with a small number of people. So each participant received 25, 100 and 250 microgram dose in 3 groups. So they received 2 doses of intramuscular injection 28 days apart. So, and then at days 43, the investigators collected the blood, and then they have found neutralizing antibody in the blood. That antibody can neutralize the virus. So they have tested it by in vitro cell culture system, that antibody can neutralize the virus, and also they did not find any adverse effect. So all the people who got the vaccine, did not show any adverse effect. So that is a very good thing. But now it is the phase trial. Phase 2 is ongoing now. Now more number of people are getting vaccinated, and then the last stage, the phase 3 trial, will start at July. So let's see. There is another vaccine. It's called live attenuated or live weakened virus. So the virus is weakened by either by mutation or by treating with formaldehyde. Actually, one company, that Codagenex USA and Serum Institute in Pune, India, they are collaborating on this live attenuated virus vaccine. But all you need, so this is the same virus, but this virus cannot produce disease, but can deliver the S protein and then make the antibody response. So you can get the antibody response without being affected or without being seriously ill. So that's the hope of this strategy. So again, just to summarize it, if you want to make vaccines, so this is you isolate a virus. That the first thing you need to do, isolate the virus and adapt the virus for vaccine. Either you weaken it by attenuation or mutation, or kill the virus or inactivate the virus, or use isolated piece of the virus. And then this is one way. And another way is you sequence the virus and then you make a blueprint of the virus or viral gene and embed it either in the plasmid or in the RNA lipid complex or in adenovirus. And then suspend all of them into the solution and then inject this and then test the solution. So now you test the solution first in the lab culture, that means the in vitro infection model of the virus, whether this is inhibiting the viral replication, and then also you inject it in mouse, in laboratory animal model, and and check whether the animal is getting sick. So if it is toxic, so if the answer is yes, then you cannot test it. So stop testing. So it is not a vaccine candidate. If the answer is no, that means animal is not toxic, then you test whether it is causing the immune response. So in the animal. So if it is causing the immune response, if the answer is yes, then you go to clinical trial. If the answer is no, then you stop testing. That means this is not a viral candidate. So in the clinical trial, it can be divided into 3 parts. Phase 1. So what phase 1 is the vaccine safe? Are there any side effects? Or it can produce induce the immune system or produce antibody. Phase 2, also you are checking the safety in more number of people and also immune response in more number of people. So if the answer is if the answer is no, you have to stop testing, for phase 1 and phase 2. If it is yes, then it will go to phase 3. So then it's a large scale trial will happen in lots and lots of people, tens and thousands of people, So whether it is protecting them from the virus. So now what is the challenge in the vaccine road? So why producing vaccine is a difficult task? I know this is pandemic, people are expecting when vaccine will come, but believe me, vaccine production of said vaccine takes time and it is difficult. So if the intensity of the COVID-19 pandemic, now these are the challenge, got significantly reduced, then it will make the phase 2-3 clinical trial extremely hard. Because what is phase 2-3 clinical trial? You are giving the vaccine and also you are giving the mock vaccine. That means people they will not know whether somebody will get vaccine, somebody will not get vaccine. It is just the placebo they will get. But they will not know who is getting real vaccine, who is getting placebo. And now they will go to the society and get infected by the virus, and then we will check whether how many of them are protected, how many of them are not protected. So without the real application in the field, it will be hard to assess the efficiency of the memory B and T cells. If there is no pandemic, then you imagine the people who got the placebo and people who got the real vaccine, both of them, nothing happened. So this is inconclusive. So you cannot make any. So when it is high time to test the vaccine, when there is pandemic going on. So that's how for vaccine trial, keeping the pandemic active is also important. So this is the one way to tell this. And also SARS-CoV-2 is mutating. If it adopts mutation in the amino acid residue in the relevant epitopes that generates the antibody, then the vaccine could reduce its efficiency. That is also a possibility. So although now universal vaccine platform for flu virus is generated, but that has not been tested for SARS-CoV-2. But that will be the future. No matter what mutation happen, so the vaccine will be universal. But the current vaccine is not following the universal vaccine platform because that will take too much time. Now the COVID-19 infection could reduce, this is another scary report that this could reduce the CD4 plus and CD8 plus T cells in some patients. So if it is reducing these two kind of T cell, then the stimulation of B cell will not happen because stimulation of B cell happen by T cell. So, and then those people may not get the full protection of the virus. So in reality, even the vaccine is there, it's not guaranteed that all people will get equally protected by the virus. So the extent of protection will vary from 30% to 70%. But there will be no, like a black and white situation will not happen. Okay, so this is the current state of the vaccine now. So 125 preclinical candidate, 7 vaccine candidate is in, this is June 13 record. Phase 1 trial, 7 candidate is in phase 2 trial, 2 candidate is in phase 3 trial, and none is yet approved. But what I anticipate that beginning of next year, there will be more than one vaccine in the market. But how efficient that will be, that only time can say. But I expect there will be moderate to high efficiency. But let's see what happens. So this is why the research should not stop, it should continue because you don't know which one will be effective. So if one is not effective, so that another vaccine will be in the pipeline so that another vaccine immediately can be made. So this is the reason. And another thing is, we should not forget that this is a zoonotic virus. This is coming from the animal. So there, we should also monitor the animal and check, what kind of virus is evolving in the animal. So this is another way. So that's why So that's why it is how it is important to continue fundamental research, in order to make effective vaccine, because knowledge should not stop, because one vaccine is not enough. Okay, so I will stop here and take any question.
Thank you.
Hello.
Thank you, sir. We are just mesmerized. It's an excellent presentation covering the whole aspect of COVID, starting from the virus, its mechanism.
Yes, I started from the very simple slide with the
Your
phylogenous
total
trait.
presentation, sir. It's really well explained and very informative. May I ask Moitri to read out some of the questions our students may have,
Yeah, sure.
answered by sir. Moitri, can you hear me?
Yeah, yeah, ma'am. 1 minute. First question is, we know that chronic inflammation majorly leads to cancer situation, and one major component of tumor environment is alternatively activated tumor associated macrophages, who
Yes.
is which is involved in promoting chronic inflammatory status. Since you are talking about plasticity of cells, so is there any possibility that in this case also that the alveolar macrophages showing alternate phenotype for pro-inflammatory condition?
So the answer is yes. You never know. So what will be the consequence? If so much malfunctioning or a lack of homeostasis is happening in inflammation, so some of them could be, if a person is before disposable, or what should I say, there are some cancer risk in that person. So this kind of abrupt inflammation can increase the risk. The answer is yes, but it will not happen for everybody. Those who are predisposed, like having a mutated gene or P53 mutation or any other cancer mutation is carrying. So for those people, this could be cancerous. But again, I told you that inflammation is a double-edged sword. So high inflammation is also, it is pro and anti-cancer also, because it also causes more CD4 plus, CD8 plus T cell, those cytotoxic T cell. One of the cause, job of the cytotoxic cell or NK cells, natural killer cells, is to kill the neoplastic cell or cancer cell. So it may be beneficial also for some people.
Thank you, sir. Next question is there any role of complement system to kill the COVID-19 by MAC formation?
Oh yes. There are role of complement system, but complement system is a part of innate immunity, natural immunity. Yes.
Sir, I have a question. Actually, just yesterday, I went through a paper and they revealed that it is in your slide also that T cell count is getting low in the COVID survivals.
Yeah, for some
So,
people, not for
how?
all. For some people, T
Okay.
cell count is low. Yes.
Okay. So, why and how it is getting low? Means T cell
So
count.
it is. There are some work on that. I don't remember. There are some work, I have seen it, that some of the viral protein is causing apoptosis of the T cells.
Okay.
So cell death, apoptotic cell death of the T cells.
Okay.
But that is not for all.
Okay. Thank you, sir.
If T cell is less, then that person will not have the full beneficial effect of vaccine.
Absolutely, absolutely.
I think
Thank you, sir. Thanks a lot.
from our students, we can mail Dr. Mazumdar from the questions from our students later also, right?
Yeah.
Yeah. One more thing is
There
yes.
there is a question I can see. Two questions, actually two questions from Dr. Biboshan Mondal. One is Glenmark is involved in phase 3 trial with favipiravir in India. According to your information, it can be a promising antidote. Is it so, sir?
Favipiravir is like remdesivir. It's an inhibitor of RNA dependent RNA polymerase. It's in phase 3 trial in India. Where?
He said that Glenmark is involved in phase 3 trial with favipiravir.
Biboshan, you can ask sir question.
Yes, sir.
Yes. So I know the remdesivir is very close to pass the phase 2 trial in here. And phase 2 trial results are very good for remdesivir.
Sir, actually, Glenmark is doing the trial, phase 3 trial with favipiravir, and the name is FabiFlu.
Yes.
The name of the drug is FabiFlu.
Yeah, FabiFlu, that originally used in Japan, that drug.
Exactly, sir. Exactly, sir.
Yes, originally used in Japan in flu virus. Because flu virus, why? Because flu virus also has RNA dependent RNA polymerase. So
Exactly.
Yes, that's true.
that's the reason. But all of them is very close to phase 2. Some of them are phase 2. Some of them in phase 2, phase 3. Now, because of the rush, now phase 2, phase 3 are getting integrated. Some company are getting permission to perform both phase 2 and phase 3 simultaneously.
Okay. Dr. Mondal, can you ask the next question?
Yeah, sir.
Your
Another
next
question is that Russia is claiming that favipiravir is doing the good result there, but they are claiming this antidote as the vaccine. Do you think so that it can be used as a vaccine?
How come a compound can be used as a vaccine? I don't see uh, uh, any rationale for that. Vaccine could be by a viral antigen. Vaccine,
Exactly.
it cannot be triggered by a compound. I really don't know how that will work. That a compound can trigger vaccine effect. I really don't know. This is against the maybe classical immunology.
Exactly, sir. Thank you.
Yes.
Thank
So,
you, sir.
any other question? But really, the more frontiers in drug discovery these days is by artificial intelligence. I just showed you 2 slides, but there are more on that. But I don't want to make things more complicated. But that's the area that where lots of promising compounds are coming out. Okay. So
Okay. Thank you,